So again, welcome. Thank you so much for joining us this morning, this afternoon, this evening. My name is Victoria Herman. I am the Managing Director of the Arctic Institute and the coordinator of the Breaking the Ice Ceiling webinar series, an incredible series that is happening throughout the spring, summer, and now fall of 2021, featuring over 20 incredible women researchers, traditional knowledge holders, Indigenous knowledge holders, practitioners, leaders, educators, artists that are sharing their expertise, knowledge, and experiences with all of you in these webinars. And today we have two fantastic speakers. I will shortly introduce our two speakers to you. I'm going to ask that we have both of our speakers present, and we will move into a question and answer session with both of them after the presentations have finished. I will be monitoring the chat throughout this presentation. If you have any technical difficulties with Zoom, please let me know in the chat or private message me. I will also ask that as our presenters are presenting, if you have questions that come up, please put those in the chat box. I will be monitoring all of those questions to make sure that we get to each and every one of them in a robust question and answer and discussion session afterwards. And again, I'll be reminding everyone of this as we are going through our two presenters. And with that, I am going to hand over my virtual mic to our first presenter, Caitlyn Bowman. Dr. Caitlyn Bowman is an oceanographer who studies mercury chemistry in marine environments, which is what she is going to be presenting today. Now, mercury levels in the ocean are on the rise due to human activities like fossil fuel combustion and gold mining. Dr. Bowman has spent nearly a year of her life at sea, spanning 12 expeditions, crossing the Atlantic, cruising through the tropical Pacific Ocean and Mediterranean seas, walking across ice floes at the North Pole, and explored the seafloor in deep ocean submarine Alvin. Now, her work across the globe is aimed at understanding how mercury connections have changed throughout history. Today, Dr. Bowman is going to present on Mercury Biogeochemistry in the Arctic Ocean. We will hear first from Dr. Bowman, and then I will ask Dr. Heather Sauyaq Jean Gordon to come on, and I will give a brief introduction before she gives her presentation. Now, Dr. Bowman, I'm going to hand over my virtual mic to you.
All right. Well, thank you for that introduction, and thank you so much for the invitation to speak today. I've been doing microplastic work for the last few years, so it's been a lot of fun to dive back into the mercury world. And I'm going to be presenting some work that I did in 2015, which seems like a really long time ago, but we're still publishing papers and we're still learning new things from this expedition that took place in 2015. So it's been a lot of fun going back through pictures and reading new papers and putting together this talk. My name's Caitlin. I'm located in Santa Cruz, California at UC Santa Cruz in the Ocean Sciences Department. And today I wanted to start by just sharing a little bit about this expedition. I spent 64 days on an icebreaker, starting in Dutch Harbor, Alaska, and going to the North Pole, and then returning to Dutch Harbor. So I wanted today just to share what that's like, what it's like to live and work on an icebreaker. I participated in the 2015 Arctic Geotraces expedition. The cruise transect that I did is outlined here in yellow. So going from Dutch Harbor to the North Pole and back. And this was a really big year for the Arctic. All of the lines in yellow that you see on this map, and we're looking at the Earth kind of top down. So this is the North Pole here in the center. The other yellow lines that you see here were other Geotraces expeditions from Canada and Germany. Geotraces is a program that studies trace elements and their isotopes in the global ocean. So the goal of Geotraces is to understand ocean chemistry, and it's this big international collaborative effort. So as you can see in this map, we kind of spread out and we look at different parts of the Arctic. So we're trying to cover as much of the ocean as we can, but you'll also notice there's some intersection between these transects. So we have these really important crossover stations where we measure the same water. So we being a US expedition, and in this case here, the German expedition. We have two different labs, two different ships, two different systems measuring the same things. And we want to make sure that we're getting results that compare well, because the end goal and the idea here is to be able to stitch together data from transects coming from multiple expeditions. And in order to do that, we have to first make sure that our data compares well. We actually overlapped with the German Geotraces expedition at the North Pole. So here we are walking across the ice. This is the Polarstern, the German expedition. I'm standing on the Healy, the US boat taking this photo. So we had the opportunity to walk across the ice and get a tour of the other ship. These were the first different humans that I had seen in about a month at this point. This is my colleague, Lars Erik Heimberger Bovida from France. And I got to, and this is my mentor, Carl Lamberg. So I got to sit down in Lars's lab and look at his data that he had been collecting for the past month in the Arctic. We don't have good Wi-Fi in the Arctic Ocean. We can't be sharing data like this over the internet. So to be able to sit down and talk about what we were seeing as we were collecting this data was a really unique experience. And I'll show a profile from this station at the North Pole. You're looking at total mercury on the x-axis and ocean depth on the y-axis. My data is in black and Lars's data is in green. So there's a lot of overlap and good intercomparison. And in doing this type of exercise, we're able to include this data in a public database. So you can go online today and you can download all of this data. Once it passes this intercalibration test, we're able to include it in this public database. We want as many people to use this data. We want you to, do with it whatever you can. So by doing this intercalibration exercise, we're able to share that. The expedition took place from August to September. This was going towards the North Pole, and this is the warmest time of year in the Arctic. So we have 24 hours of sunlight. This is when ice is melting. We turned around at the North Pole and headed back to Dutch Harbor, Alaska, in September to October. So now we have the return of nighttime, temperatures are dropping, and the ice is refreezing. So from a chemist perspective, this was a really important time of year to capture. So we're going through open water and melting ice, and we're coming back through freezing ice. And I'll show you why that's important for the Mercury cycle. We reached the North Pole on September 5th, and we celebrated Christmas. So the whole inside of the ship, we had stockings and Christmas trees. I got my picture taken with Santa. We had a nice celebration at the North Pole. It was a celebrated occasion by the US Coast Guard. So we were on the Coast Guard cutter Healy. This was the first time that a US surface ship has reached the North Pole alone. Normally, they go with another ship, usually from a different nation, to help break the ice and get to the North Pole. So it was celebrated by the Coast Guard, and a testament to the vehicle and the skill of the captain, and kind of touted as this achievement. But as a scientist, I had kind of a pit in my stomach, because I don't think we should be able to do that. We're seeing this big shift in the Arctic Ocean from thick multi-year ice to more thin first year ice. And I think that's part of the reason we were able to do this expedition with just one ship. So kind of a bittersweet moment reaching the North Pole. This is where I lived. So we have bunk beds. I often say that going to sea is like being in summer camp. This was the coffin bed. It's usually a couch, but the third person, we had so many people on the ship that each room had 3 people. There's a bathroom in between here and 3 other women on the other side sharing that space. We had a science lounge to ourselves. There's no internet or TV or radio or anything like that. We watched Bob Ross DVDs for 2 months, and the Coast Guard did a really good job at building community on this ship. So once a week, we would have some sort of event, whether it was sumo wrestling. Here we had a big talent show. So both science party and crew members were singing and dancing, and we had a lot of fun. It's 64 days, over 2 months at sea, and there's a lot of time before and after the expedition setting things up and tearing things down. So many of us were gone for 3 months. So this type of community building was really appreciated. At the end of the expedition, we were able to see the Northern Lights. Being out on a boat where you have no light pollution, it was just a really incredible experience. We also had some nice interactions with wildlife. There were 150 people on board this giant icebreaker. Everybody had to have a pager. And you had to check in the morning and the afternoon. So you had to sit down on a ship computer and you had to log in the morning and the afternoon to make sure you were accounted for. It's to make sure we didn't lose someone overboard or someone down in a deep layer of the ship. But we also use these pagers for emergencies and then for wildlife sightings. So here, this is a day where I was working in the lab and I got a page that there was a giant polar bear that I should go outside and see. So we had a few different bears visit us, very curious, kind of stick their nose in the water, check us out, hang out for 20 minutes, and then carry on. Okay, let's look at the work. As a chemical oceanographer, I mainly look at seawater. Here we're collecting seawater with what's called a rosette. There are long go-flow bottles. So it's a long cylinder that's open on the top and the bottom. We lower the rosette to the bottom of the ocean. And as we bring it up, we trigger those bottles to snap shut. So we are collecting water from the surface of the ocean to thousands of meters deep. And we're looking at profiles. So how does the chemistry of the ocean change from the surface to the bottom? We have all these different layers in the ocean water column. We also look at particles. So imagine pouring seawater through a coffee filter, you would collect a bunch of gunk. Those particles are really important to marine chemistry. So we have a team here that is setting up this pump. So it's a battery powered pump with multiple different filter heads. It weighs about 100 pounds and they go to the edge of the ship, they attach it to a wire and they lower it down in the ocean. It sits for a few hours pumping water through those filters. And then we also look at surface water, but we don't want the surface water surrounding our ship, our big metal rusty ship. So for surface samples, we try to move away from the icebreaker. Here we have a small boat operation. So this team is moving away from the boat to collect uncontaminated surface water. The go-flow bottles here, which contain seawater, are transferred into a really special laboratory. This laboratory, it looks like a shipping container on the outside, but on the inside, it has HEPA filtered air. So low particle air. Everyone working in here changes their shoes before they come in. They're wearing gloves. And what you see here is the go-flow water being drained into sample bottles. And there's stickers on these bottles. So every bottle has a special code, and that code gets attached to every single sample that comes from that bottle. So this particular sample, this particular water sample might be analyzed for 12 different things. And by having this tracing system, I can measure this water for mercury and I can compare it to a dozen other trace elements. And that's going to help me understand more about the chemical cycling happening in this water, where mercury is coming from and where it's going. So we have this incredible system. These girls work so hard filtering thousands and thousands of liters of water. We're also interested in sea ice. So here we're collecting clean ice cores that will melt to look for trace elements. There is also a student on board looking at DNA and plankton in the ice. We look at the water directly below the ice as well. So this was my job. You're looking inside an ice tent here. And this incredible team is pumping water from just below the ice, and we're filling all of these sample bottles for different analyses. So my job was outside, organizing these bottles, passing them inside, and then once they were full, I would load up this sled and walk them back to the ship, pass them off to the scientist that would either preserve them or start analyzing them. So really fun job working on the ice. For mercury chemistry, I look at a couple different gaseous forms of mercury. So if you opened a can of sparkling water and left it out overnight, it would be flat the next day. All of the carbonation escapes to the atmosphere. Same thing happens with mercury in seawater. If you want to look at these gaseous mercury species, you have to analyze them as soon as that water sample comes up. So we pack up our labs at UC Santa Cruz. This is Dr. Allison Agather, who was doing her PhD at the time, and she was at Wright State University. So we pack up the lab at UC Santa Cruz and at Wright State, shipped it to Alaska, and then we rebuilt our lab on the ship. So we have this special shipping container lab that Allison and I worked in. And as soon as that seawater is coming out of the go flow into a sample bottle, it's brought directly into the lab and we start processing and analyzing that right away. This is the inside of one of my detectors. Why are you seeing that? Well, the instruments that we use in a chemistry lab are not designed to be run every day for 12, 15, 16 hours a day. So there's a lot of maintenance and a lot of troubleshooting that comes with running your lab at sea. It can be very stressful. There are a lot of long days. And then I also was looking at DNA in the Arctic. So there are some important genes, there are some genes that are important for mercury cycling. So back in the lab at UC Santa Cruz, I had a little bit more lab work to do, but most of the analyses were able to finish on the ship. Okay, so I want to talk a little bit about mercury toxicity, the motivation for doing this work. Humans are exposed to mercury through seafood consumption. And the most important point to remember here is methyl mercury accumulates in seafood. Inorganic mercury goes into the ocean. So once inorganic mercury is deposited to the ocean or the environment, there's a lot of chemical cycling that happens, and that can produce an organic form of mercury called methyl mercury that's able to bioaccumulate and biomagnify in marine, aquatic, freshwater, terrestrial environments. So in high trophic species like albacore tuna and swordfish, those species have higher concentrations of mercury. Marine mammals have higher concentrations of mercury in their bodies as well. The map on the right is showing studies with a strong connection between seafood consumption and mercury concentration in the body. So these studies are looking at mercury in hair, blood, and urine, and that correlates directly with diet. In the Arctic, marine mammals and pilot whale consumption has been linked to higher mercury levels in humans. And we see this in other populations across the globe, including the Mediterranean, the Great Lakes, Japan. So the diet of a community is a good indicator of mercury concentration in the body. This map is showing different mammals in the Arctic that have varying degrees of mercury-mediated risk. So for this study, these animals had mercury concentrations in the liver measured. Anywhere you're seeing orange and red, these are high concentrations in the liver that could cause adverse effects in these animals. Most marine mammals in the Arctic had no risk or a low risk of health effects from this mercury exposure, but certain populations had a high risk. Polar bears in Alaska and the Canadian Arctic had high mercury concentrations. And then other populations of toothed whales. These are whales that are eating other fish like pilot whale, narwhal, beluga, and hooded seals had high concentrations of mercury in their body. This is another study that looks at long-term monitoring of both marine mammals and seabirds. And these studies are looking for a link between global climate change, induced changes, and mercury concentration. And in many of these studies, it was found that increasing mercury concentrations can be correlated with climate change. This is things like changes in trophic interactions, prey availability, diet, foraging region, foraging time. So the changes that are happening in the Arctic in some populations are resulting in higher concentrations of mercury. It's important to think of mercury as one of many stressors that these marine mammals in the Arctic are facing. Mercury is not the only problem. Mercury is contributing to the many problems and the many stresses that these animals have, including infectious disease, climate change, increasing human activity. So mercury on its own is harmful, but it's also contributing to a larger problem. Mercury Biogeochemistry. So biogeochemistry is a term that describes the cycling of chemicals through the environment. With mercury, it's very complicated. Mercury comes from both natural sources like volcanic emissions and human sources like coal combustion for energy. And once mercury is released, it's released as a gas and it can stay in the atmosphere for 6 to 12 months. So there are no volcanoes and coal-fired power plants lining the Arctic, but mercury emissions from lower latitudes are transported with air masses up to the Arctic, and that's one of the biggest sources. So mercury gas is oxidized in the atmosphere to divalent mercury and deposited in the ocean. Some of that is reduced back to gaseous mercury and can go back into the atmosphere. So in some parts of the world, the ocean is releasing mercury gas to the atmosphere. And at the same time, we have this deposition of mercury. And this divalent mercury can then undergo several different chemical changes. This green layer here, so this graphic is representing the ocean, and we have this green layer that represents phytoplankton. These are shallow waters where there's enough sunlight for photosynthesis, and that photosynthesis produces oxygen. But once this biomass begins to die and sink, it's decomposed by microorganisms. So these microorganisms feed on that organic matter, and in the process, they lower the oxygen concentration in the water. This region of low oxygen in the ocean is where we typically see higher concentrations of methylmercury. And methylmercury, again, is the species that accumulates in food webs. So these microorganisms that are active in this part of the ocean consuming oxygen can also produce methylmercury. There's a gene cluster found in certain bacteria that can produce methylmercury. There are other microbes with a whole different set of genes that can degrade methylmercury. So microorganisms play a really important role in mercury biogeochemistry in the ocean. It's even more complicated in the Arctic because now we have sea ice. So salt is not incorporated into sea ice. When the surface layer of the ocean freezes, the actual solid ice is fresh water. This can release halogens into the atmosphere like bromine that can oxidize gaseous mercury and deposit it to the surface of snow and ice. So they're called atmospheric mercury depletion events. They're more important in coastal environments. When seawater freezes, it also rejects brine. So we get this really salty layer that underlies the ice. And when the ice melts in the springtime, we have this layer of fresh water on the surface ocean. And then underneath, we have this really, really salty layer. So the Arctic Ocean is very stratified. Think about mixing salad dressing with oil and water and vinegar. You would see a settling of those 3 different layers with 3 different densities. So the Arctic Ocean water column is like salad dressing, where we have these 3 different stratified layers. They have different densities due to different salt concentrations. New discoveries and future trends. So what have we learned since 2015, since 3 different nations went up into the Arctic, and there has been a lot of work outside of Geotraces as well. So I want to talk about new discoveries, but first say that my role as an oceanographer in understanding mercury biogeochemistry in the Arctic is to help quantify sources and sinks. So we have an idea of where mercury is coming from, how it's getting to the Arctic. My job is to tell you how much, to quantify each of those sources, to understand where mercury goes once it enters the Arctic Ocean, and to understand the chemical cycling of mercury. The end goal is to use that data in mathematical models that can then predict how mercury will change in the Arctic Ocean under different climate change scenarios and under different emission scenarios. So if we keep emitting as much mercury as we are today for the next 30 years, what will mercury in the Arctic be like? Or under different warming conditions, what will mercury in the Arctic be like? So the end goal here is to feed this information into a model to help us understand how mercury will be affected by change happening in the Arctic. Unlike other ocean basins, we see high concentrations of mercury in the surface ocean. And this is due to accumulation of gaseous mercury under sea ice, which has been observed previously in the Arctic. This is our transect showing gaseous elemental mercury concentrations on a color scale. And in white, we have sea ice cover. We broke through that ice to sample water, and what you'll see underneath the ice is higher concentrations of this gaseous elemental mercury. So the ice sort of acts as a cap. I mentioned previously that gaseous mercury can evade back into the atmosphere, but it accumulates underneath the ice cover. There's a big shift happening in the Arctic from thick multi-year ice. This is ice that doesn't freeze during the spring and summer. It doesn't melt during the spring and summer years. And we're shifting towards first year ice. This is ice that freezes and thaws each season. So we now have predominantly first year ice, where before 2011, we had mostly multi-year ice. On the left, this is a paper by Brian Demento, who is working on this expedition. And he's again showing gaseous elemental mercury in surface water. And the color here that you're seeing, the blue, represents sea ice cover. So gray in this map is showing open water. The darker blue shades are first year ice, very young ice. And the lighter shades are older multi-year ice cover. So as you can see, when we get closer to the North Pole and we get into this lighter color, this older ice, we have higher concentrations of elemental mercury in the surface ocean. But when we get to first year ice that's been around 2 to 3 years, we have lower concentrations of elemental mercury. So perhaps with decreasing ice cover in the Arctic Ocean, we will see elemental mercury evade to the atmosphere and lower concentrations of mercury in the surface ocean. However, another study that looked at ice from the US and the German transect found higher, significantly higher concentrations of methylated mercury in first year sea ice compared to multi-year sea ice. So this thick multi-year sea ice has lower mercury concentrations than the first year ice. And again, we're seeing a shift to predominantly first year ice in the Arctic Ocean. So first year ice, if it has higher methylmercury concentrations, we get sea ice algae underneath the ice that could be exposed to that mercury. And we could potentially see increases in mercury going into Arctic food webs as we shift to more first year ice. We sampled a current called the Transpolar Drift, and this was characterized by Lauren Kipp, who's here today. The Transpolar Drift is the current that transports water from the East Siberian shelf. So we have freshwater input from rivers, we have shelf water that over in the world, but it receives 10% of riverine discharge. So we think that a lot of mercury is lost closer to the source. It's either reduced and evades to the atmosphere, or it's scavenged by particles and settles out on the continental shelf. We don't see a strong connection between riverine inputs and mercury concentrations all the way out in the middle of the Arctic Ocean. However, one of my big roles in this project was looking for mercury cycling genes. So on the right here, you're looking at particulate and dissolved methylmercury concentrations in the transpolar drift. So here's the surface. And then on the left, you're looking at the abundance of different mercury cycling genes. So here we have HGCA-like genes, which could potentially be involved in mercury methylation. We have a greater abundance of MER genes. These are genes that can decompose methylmercury. So greater abundance of these genes within the transpolar drift. The transpolar drift is also pushing carbon and nutrients, and that could potentially be providing substrate or food for these microorganisms that are important to the mercury cycle. So really this finding just makes it more challenging to understand and predict methylmercury concentrations in the Arctic Ocean. With the presence of these genes, it kind of complicates the abundance of these genes could be affecting methylmercury production and degradation. Finally, this is the first couple profiles of methylmercury in the high Arctic. And this paper shows, this was published in 2015, the year that we all went to the Arctic, and it shows this high concentration of methylmercury in relatively shallow water, so at 200 meters. And in the beginning, I mentioned we normally see high methylmercury in the ocean where there's low oxygen. But in the Arctic, and this is something that was consistently observed in the US Geotraces, and this is a plot from Canadian Geotraces as well, we're seeing this relatively shallow peak in methylmercury in oxygenated water. And the hypothesis put forth in this paper was that I mentioned the Arctic water column is like salad dressing. We have these stratified layers. And so sinking particles coming through this freshwater layer kind of slow down once they have hit the high density halocline. And the slowing down of those particles allows microorganisms to feed and potentially produce methylmercury in this more shallow depth. That's the running hypothesis. Our observations are all finding this relatively shallow peak in methylmercury. This is the Canadian Geotraces, so the Canadian Arctic Archipelago, and this is US Geotraces, where we have shallow peaks in methylmercury. Arctic warming could lead to increased methylmercury production. So we could have increased stratification with warming temperatures in the Arctic that could potentially lead to more methylmercury. Big takeaway from this discovery: in lower latitudes, we use something called apparent oxygen utilization to predict methylmercury concentrations. So we're kind of assessing how much respiration is happening to predict methylmercury. But that's not going to work in the Arctic. So just knowing the amount of mercury that's there and knowing how much respiration is occurring is not going to help us predict methylmercury concentrations in the Arctic. So that was a really important finding in terms of working with modelers and figuring out how we can predict methylmercury concentrations in the future. And with that, I want to just acknowledge all of the incredible women that I got to work with on this expedition. I am one of 20 female scientists that was in the Arctic Ocean out of, I think, 51. So almost half of the scientists on board were women. These are graduate students, professors, career researchers, wives, mothers, women that go to sea for months at a time to understand marine chemistry. So I'm very lucky to get to work with all these incredible women. Great, thank you.
Thank you so much. If everyone could join me in giving a huge virtual round of applause to Dr. Bowman. Thank you for such an incredible presentation and a great ending. We are going to now switch to our second amazing presenter. I'm going to hand over my virtual mic to Dr. Heather Sauyaq Jean Gordon, who was born and raised in Homer, Alaska. She is Inupiat and an enrolled tribal member of the Nome Eskimo community. Dr. Gordon has received her BA on Race and Ethnic Studies, an MS on Sociology, and a PhD from the Indigenous Studies program of the University of Alaska Fairbanks, where she not only wrote a dissertation, but produced a 20-year roadmap for the community outlining the results of her research. Incredibly impressive. She will be presenting today utilizing self-determination to achieve sustainability and well-being. Dr. Gordon, I will hand over my virtual mic to you.
All right, thank you so much. Yeah, so thank you so much, Dr. Bowman, for your presentation. I do have quite a change of gears as my work is much more social than physical, I would say. So a little bit we'll be learning from each other today, which is wonderful. So I'm going to be presenting on one part of the research, talking about how ethnographic futures research is a really great method to work with our Indigenous communities to develop sustainability indicators. Oops. I just have to give a brief disclaimer because I do, since I got my PhD, I do work for the Administration for Native Americans now. So I'm just required to say that my presentation is just about my dissertation work, which ended in 2019. I've started working for the Administration for Native Americans in after all my dissertation work was done. So this is not at all related to any of the work I do now. This is my dissertation work. Okay, so I'm going to give you a little background about myself. I'm going to talk about the project site and participants I worked with, and then I'm going to give a little talk about the Indigenous methodologies and ways to engage with Indigenous people in research in a good way. I'll get a bit into ethnographic futures research as the specific methodology I used in my dissertation, and then we'll talk a little bit about how that works so well to identify sustainability indicators. That was a great ending, Dr. Bowman, talking about women in research. So I actually want to start out with that same trend, I guess I would say. So my own background, I'm Inupiaq and Western European. I'll introduce myself in my language very briefly. So Uvalawata, Paglagisi, Atiga, Heather Sauyaq Jean Gordon, Inupiaq Siniga Sauyaq, Akaga, Sue Elizabeth Gordon, Apaga, William Lewis Strutz. So good morning, and I welcome you. My name is Heather Sauyaq Jean Gordon. Sauyaq is my Inupiaq name, and my mom is Sue Gordon, and my dad is William Strutz. In this pictures, you will see my family, the Native women in my family who have really led to the work I do due to their experiences that they had in life. So my great-great-grandmother, Ella Kaguna, is on the left. Her daughter, Margaret Cecilia Becker, and then my grandmother on the right, Mary Jean Kaguna Yenny. The women in Native women in my family really had a rough history education-wise, and with racism in the Jim Crow era in Alaska. That's a period my grandma lived through, where Natives had to go to different schools than white children, and sit in different parts of the movie theater, drink different water fountains, very similar to some of the practices that were happening in the American South, actually. My great-grandmother, Margaret Cecilia, attended boarding school, and it was Holy Cross boarding school. It wasn't a very great experience. Her experience was so extreme that she actually did not ever speak Inupiaq to any of her children. So the language really ended in my family with my great-grandmother. Okay, so now I'm going to get a little bit into how I engaged with Alaska Natives in a good way in this research project due to a very traumatic history we have with assimilation and colonization. So the population, the people I worked with were from Ninilchik, Alaska. So this arrow is pointing to the Kenai Peninsula and to the community of Ninilchik, which is very small, under 900 people year-round. There's a bit of a swell in the summer as people come up for their summer homes. The community is a native Russian community. So in the late 1700s, when many Russian men were coming to Alaska for the furs, hunting different marine mammals, they often married native wives. And so Ninilchik was founded as actually a retirement community for those Russian men. So the descendants of these people from the 1800s are quite a mix of different tribal peoples in Alaska, because the Russians were reaching many different parts of Alaska, down on the right in the southeast, working with people from there, the Kodiak area, the Sugpiaq people, Yupik people in like that darker blue area, and Dena'ina people kind of that whole kind of central reddish area. So there was a really diversity of native people that ended up in Ninilchik. And now the Ninilchik village tribe is the local tribe in the community. They have about just over 900 members, but that would be worldwide. They're only about 20% of the actual population in Ninilchik year-round. And I talked to both tribal and non-tribal members for this project, because the Ninilchik village tribe really stated that they wanted the results of the project to be able to be used for them to apply for funding, government funding. And the government funding they do is often not Native specific, since they serve both a Native and non-Native population in their community. And so the people I talked to were parts of all different jobs and positions in the community, really ranged in age from the 20s to the late 70s. I talked to tribal members, non-tribal members, people who worked for the tribe, people who didn't. And so there were 30 participants in the project. So I just want to start about, how do we approach working with Indigenous people? And in this project, that's what I was doing. So you'll often hear, if you're in Indigenous communities, people will talk about doing research or starting something in a good way. And that in a good way is referring to, honoring our spirits and traditions, honoring our elders and our ancestors. So to me, today, to start out my presentation in a good way, I wanted to start with introducing myself in what little of my language I know, and introducing the Native women that really inspired me to do the work I do to build well-being in Indigenous communities. So there's a history of historical trauma and research abuses in many Indigenous communities. I don't know if you're that familiar with, starting from the historical trauma present from assimilation and colonization practices that people experienced, whether it was forced assimilation, boarding schools, not being allowed to speak their languages, removal policies, criminalization of culture and outlawing indigenous religious practices. So there was a variety of things that didn't allow indigenous people to practice their culture, language, or way of life, and even took children from their homes and their parents to be put in boarding school. So this resulted in some painful things in our communities that has led to different ways this is being manifested today. So if you look at research by Dr. Braveheart and many other researchers, you will find that historical trauma can be seen to be manifested in ways today through such things as like substance use, suicide or suicidal ideation, the loss of native language and culture, some issues with poverty in many communities, and trauma and also domestic violence. So I wanted to talk about how those issues, which we can say, were very historical, potentially, although they're still within the last 60 years, that there's kind of been continuous issues in Indigenous communities around research abuses. So in Alaska specifically, I wanted to just mention the baro alcohol study and the thyroid function study, where research was done without really being done thinking of putting the participants first. In the baro alcohol study, it was done in the late 70s, and there was an agreement between the Native leaders of the community and the researchers to do this project and explore how alcohol use was affecting the community. However, the big problem came when the researchers gave their results to the press before talking to the community about the results. So the New York Times actually ran a front page story in 1980 talking about the plagues of alcohol in Native communities in Alaska, and actually quoted one of the researchers saying, "We don't see the Eskimos surviving till 2010 to 2015. And this researcher actually was quoted as saying, "This is not a collection of individual alcoholics, but a society which is alcoholic and therefore facing extinction." So, I mean, imagine if you read that about your community and your family, and not everyone was an alcoholic. It was not a society of alcoholics, so this perspective, the way it painted the community and Alaska Native people, was really stereotyping and marginalizing the people in the community. So due to this historical trauma and these research abuses, we really try to engage with an asset-based, strength-based research approach through participatory research when we're engaging with Indigenous communities. Historically, when the anthropologists were the main researchers going out into Indigenous communities, they were seen as the experts on our people. Because they were the ones who wrote books about us and things like that. But now you'll find when research is being practiced in a good way, going into an Indigenous communities, the community members are the experts on their community and on their knowledge and traditions. And that, as a researcher, you're there to learn and to see, how you can learn from them and how you can give something back to the community as well. So really looking at using participatory methodologies. I'm going to talk a little bit about an Indigenous relational theoretical framework that came out of my master's work. And also some of the benefits of having that asset-based approach instead of a deficit-based approach. It not only addresses that historical trauma and research abuses, but also provides hope for the future. If we talk about things as not, this is a community of alcoholics, but the current condition in this community has, this percentage of people utilizing alcohol. There's hope for the future and change and growth. And it really lets us avoid stereotyping and marginalization. So instead of, putting the emphasis on how many people abuse alcohol, instead looking at what are the protective factors for all the people not using alcohol. How is culture being used to educate the youth, to keep them, on a good path, interested in their future and positive about their life? So really emphasizing that well-being and recognizing the self-determination and sovereignty in the Indigenous people, that they have those rights and that they are unique governments and unique peoples in their own right, whether they identify as tribes or communities or villages as well. So having that understanding of sovereignty and self-determination when you're working in these communities. And especially this is important when you're doing research around health, because health research is often very deficit-based, comparing the rates of suicide from one community to another, or the rates of heart disease in one ethnicity and another. But those deficit approaches can be very marginalizing and stereotyping, as you can see what happened due to the baro alcohol study to the Alaska Native Inupiaq community in Arctic Alaska. So the framework I developed from my master's work is this 8-point model as a way to build a trusting relationship that can be mutually beneficial in working with Arctic Indigenous communities. This project, I worked with North American Arctic researchers to get one perspective, and then I talked to Inuit Greenlanders through interviews and focus groups to get another perspective, to have both sides, to seeing what's important and what is underlying that we can do to work for to benefit everybody involved. And it really came out that trust was the key aspect that you're searching for in your relationship building, and that it's approached in this specific way. And so I used this model for my dissertation, working with an Inilchik village tribe. I started out working around knowing community history, researching the community, so I knew if past research abuses had happened there and what things I should avoid. I have a local contact through a fellow student in my PhD coursework that really served as a liaison between me and the community. And through him, I was able to be introduced to the tribal leadership and start building a relationship with them. I visited the community for a face-to-face meeting to discuss the project, the research questions, what they were interested in knowing. Really recognizing that face-to-face communication is so important in Indigenous communities. I worked to really respect the interests of the tribal government in Inilchik, as they wanted me specifically to work through the liaison that I was that student that I had been working with in class, because the tribal government is burdened with many activities and things they're doing. So it was easiest for them to appoint one person to be the main person that I would work back and forth with, and he could bring them my perspective and me their perspective. There's this aspect of having good manners. This really came out of the Indigenous respondents to the project, talking about, as much as you may be employing institutional ethics, you might have done an IRB review, IRB approval, but are you engaging in the manners of the community you're working in? Which is such a great thing to have those local contacts prior to working in the community, so they can teach you how to act. And help avoid, a blunder that you might not even do on purpose, but that could be seen as offensive in the community you're in. Really valuing co-production of knowledge and exchange of knowledge through the research project. Even if you're a physical scientist, you'll often see Dr. Matthew Sturm, who is an ice scientist, that he, even though his work is physical and he's working on the ice, he still takes the time, if he's in a community, to go into a classroom and present on how you do ice science, getting young people exposed to something they wouldn't otherwise be exposed to, and see the potential of a scientific career in the Arctic. That can be done. So, finding ways to give back, even if it's not directly a part of the project you're conducting. And then, giving back the data in a way that's actually useful for the community. This just shouldn't be only talked about at the end, of course, but something from the very start of the project. In Inilchik specifically, like I said, they wanted to use the results to apply for grants. So they were very intent on me following through with publishing my dissertation work so that they could cite articles in their grants. So utilizing that model, I worked with the ethnographic futures research methodology, recognizing that EFR is traditionally a Western model based in anthropology and collaborative research. However, adapting it to the Indigenous context through that relational framework, emphasizing the giving back, emphasizing the 8 steps to trust building, as I talked about in the previous slide. So I'll talk a little bit now about ethnographic futures research and how it has 3 main scenarios. So you have an optimistic scenario, which is a number 90 on a scale of 1 to 100. You have a pessimistic scenario, which is number 10 on a scale of 1 to 100. And then after you've given those 2 scenarios, you can kind of orient what's the most likely direction the community will go in. So I was talking to community members about, describe your community 20 years in 20 years. So for the project, it was 2018. So I said, in year 2038, looking back over the last 20 years, how has your community arrived to the optimistic way it is? You have a community with sustainability and well-being. How did it get here? And then the pessimistic one, you're at a 10 on 1 to 100, looking back over the last 20 years, you're in the present in year 2038. What does it look like? So, adapting this method to be appropriate with Indigenous people was key to the project. And so it had a participatory, collaborative approach, as the scenarios are really a storytelling aspect which draws on Indigenous, the way oral history has been passed down. We also did focus groups, which were run more like talking circles, where, I made sure to bring food and cook food and really host people, and start by introducing myself and looking at the project in that good way. So after doing the optimistic, pessimistic, and then most likely scenario, then the 4th part of the interview, the interviewee talks about how to reach the optimistic future and what their specific role in reaching it is. So not only is this an asset-based approach, really looking at where the community can go, but it's also an optimistic approach, which actually is shown to lead to well-being, even in the participants just engaging in the interview. I'm just putting forward that thought process. And I had people after the interview be like, wow, I didn't even think about what I can be doing now. I'm raising my granddaughter. I'm going to make sure to teach her gardening so she knows how to provide for herself later. So it was a really positive approach to the community. And it really had that asset-based, looking at, where can the community go to achieve what it desires. So I interviewed 30 different people in the project to get a perspective with a lot of different viewpoints for the community. So the sustainability indicators, these are just a selected few. So these came out of analyzing those 30 interviews and the focus groups as well. So the sustainability emphasized, people really talked about economic, social, and ecological components of sustainability, which you'll often hear about in the definition of sustainability as well. However, when asked, could you define sustainability for your community? That was really a difficult question for people to answer. I would get something along the lines of, residents should be healthy, or that whatever we're doing isn't going to make it hard for my future generations to survive. So I was like, how can we really get to the definition of sustainability? And these indicators, what they came out of was those detailed the detailed scenarios. So as someone's explaining that optimistic future, they're breaking down what that looks like in infrastructure in their community, what that looks like in the education, what that looks like in their environment. And then when they talk about the pessimistic future, they're really detailing what isn't there. So I was finding that these indicators of what the community wanted to see in their future were really coming out of the in-depth stories they were telling me. So some economic things, they really stress growth in small businesses and in tourism in the community. And the Milkshake is very small, so they're trying to find a way of how to bring more economic spending into their community. Some social aspects were increasing affordable housing. Even though it's a small community, there's always a lack of housing, and people are having to live in different places. So the Ninilchik historic land base actually extends from Homer to Kasilov on the Kenai Peninsula. That's quite a number of miles apart. Ninilchik is about 40 miles south of Kasilov and about 40 miles north of Homer. So I had grown up in Homer, which was on actually Ninilchik traditional tribal land. So some other really important things they wanted to see in the optimistic future in their community was seeing youth returning to the community to live. There's a lot of talk about outmigration. As young people get further education and skills, how can they be a part of the community that they were grown up in? Are they able to return back after getting those higher educational degrees? What's there for them to be able to work and have money to support their families? Ecologically, people were really interested in sustainable resource management. And one of the things that came out of the work is the importance of including Indigenous voices in sustainable resource management that's often managed at the state and federal level without a lot of traditional ecological knowledge or local Indigenous knowledge being included. So there's this quote about the future by Cornish that says the main point in thinking about the future is to change it, to make it better than it would be without deliberate choices and actions. So this ethnographic futures message really looks at what are the deliberate choices and actions that the community wants to make to have a sustainable community. And by engaging them in these scenarios, the optimistic future showed the community members described to me what they could achieve and what were the possibilities of having a thriving community. The pessimistic future really addressed what communities would not be doing, what was lacking in their community. So as much as sustainability is often a difficult concept to explain, allowing people to tell stories, which is a more traditional approach to understanding Indigenous knowledge, was a great way to understand sustainability in the community of Ninilchik. So I just had some sources, and then I just wanted to say thank you so much for the opportunity to speak today. I'm thrilled to see all the female women scientists in all, social, physical, all the different realms as well. I had support from the National Science Foundation and Mellon Foundation for this work, so I just wanted to say thank you as well. So Victoria, I will turn it back over to you.
Thank you so much for an incredibly thorough and thoughtful presentation. I know I was taking lots of notes throughout it, and I hope everyone else here was as well. We have about 25 minutes for questions from all of you. I am going to bring both of our speakers back onto our virtual stage. And we already have some questions that are coming in. Our first question is from Elizabeth, who I am going to ask to come on to ask her question.
Sorry about that, to find all the buttons. This is for Caitlin, or Dr. Bowman. And my question, I noticed you mentioned that you had some wildlife encounters when you were on the ship. And I do a lot of work on polar bears and was curious about where those interactions happened and if anyone kept any records of them. If it was in the Arctic basin, that would be really interesting because there's really not much work that's been done on polar bears up there.
Yeah, I think that's something I could figure out and pass along to you. I don't remember 2 months of working 24-hour weird shifts. I can't place them, but I think just through, I had a blog during the expedition. So I think through the blog and then dates associated with the photos, I could definitely pass on that information to you. Maybe type your email in the chat. And
thank you.
Great, and we can definitely facilitate that in our follow-up. We have another question come in from Sasha for both of our speakers. So the first question for Dr. Bowman, can you discuss how the migration of fish species further north due to changing water temperatures might impact mercury concentrations? Do different fish have different potential for bioaccumulation?
So I'll answer the second part first. Basically, the more fish that a fish has eaten, higher concentrations of mercury in the body. It's not necessarily a different potential for accumulating mercury. It has to do with what trophic level that fish is feeding on. I love the first question. I don't have the answer to that, but that's really, really interesting. Maybe you noticed in the image that I showed with all the different Arctic marine mammals and whether or not they had high or low mercury concentrations in their liver, there's not really a clear pattern there. We had these kind of isolated communities of marine mammals that had really high mercury concentrations in the body, and we're still trying to understand why. There are so many different factors with river input, with sea ice, with the way the ocean currents move around the basins, kind of the circular motion. There are a lot of different factors that affect regional variations in mercury. So I think that's an excellent question that we don't have the answer to yet, but hopefully someone's looking into that.
And maybe one of our early career participants on this call has an idea for what comes next. The second question that Sasha had was for Dr. Gordon. Can you discuss principles that should be followed for reaching out to Indigenous researchers for collaboration on non-human based research? How can collaborative research be fostered with Indigenous researchers for topics that are not necessarily directly connected to community well-being?
That is a fantastic and comprehensive question. So I'm going to first say that actually, can you clarify a little bit? Are you looking for building relationships with Indigenous researchers, or are you talking more about that your work will be done in an Indigenous community where you might be interacting with community members, or are you actually seeking the engagement of Indigenous research partners? Oh, with Indigenous researchers. Okay, thank you. Okay, so there is some great things being done I'm not sure where you're based. There's in the US through the National Science Foundation, there's actually some different meet and greets they're actually doing right now through the Office of Polar Programs, through the IARPIC working group, and they're trying to make a space for researchers to come in from the social, physical, different sciences as a way to get to know each other and even create that space. Because I think so often we're working in such different circles. Like today, this was really a unique opportunity. I never hear about the type of work that Dr. Bowman does. So this has been really wonderful to speak to you all. So there are ways that the National Science Foundation is kind of trying to help connect people. That's really stemming kind of out of the lack of co-production that has been a little bit problematic in some Arctic research. Oh, I think I'm sorry, my computer was dying, so I had to plug it in. Let me just move, I think. I am on a competing meeting call with someone else right now. Okay. Okay, so other great ways to build the relationships that I found in my own work. The Arctic Frost Network was an early career scholars network that connected people who did work in the Arctic, and it spanned, from permafrost researchers to glaciologists to those of us doing social science. So I think really looking at these research networks, even the research networks that are funding these talks, that those are a way to join and kind of get to know people who do different types of stuff. another thing that I often do to connect, if I'm interested in working in a community, you can look at who has maybe published research out of that community before. Like specifically, I was looking kind of at the Marine Mammal Protection Act and how it's working with Indigenous people. And I found some research by an anthropologist in Alaska and by a sociologist at UCLA. So kind of looking at the history of work that have been done in that community or area can also kind of expand your connection to different researchers as well.
Thank you so much. We have another question I am going to ask Delove. Do you want to come on and ask?
Yes, I hope you can hear me. Yes, so first of all, I would like to thank Dr. Bowman and Gordon, if I pronounce the name right. If not, forgive me, for the powerful presentation. And I appreciate you setting an example for more females to join the sciences. But my question is for Dr. Bowman. I want to know which form of mercury you recommend for an incubation experiment, either between methyl mercury chloride or mercury chloride. Which of these two forms will you recommend for an incubation experiment?
When I do incubation experiments in seawater, I start with mercury or methyl mercury chloride, and I add it to seawater. So I dilute the mercury with seawater, and then I let it sit for 24 hours. And that gives the mercury a chance to equilibrate or to bind with other ligands in the seawater. And we think that that's a more natural form of mercury to be available to microorganisms. If you also want to drop your email in the chat, there's a really good paper I can send you that explains that. But we do these incubation studies with mercury where we can add mercury isotopes, or we add a known amount of mercury to water, and then we watch how it changes. So that's a really important question, kind of how to start that type of experiment.
All right, thank you. So I'll drop my email. And then...
I'll send you the paper.
All right, thank you.
Yeah.
Great, thank you. I'm going to ask Elizabeth to come back on and ask your question to Dr. Gordon.
Hi, thank you. I think this follows on really well to the other question about networking. I've actually been looking at some of Dr. Gordon's work that I found on ResearchGate after reading other people's work. So it was great to see your name come up on this presentation. My question for you is around co-production of knowledge, and it's kind of the thing that everyone, including myself, want to aspire to. It's a much different question of doing it is very challenging, and I feel like most of the examples I've seen so far have been kind of indigenizing Western science or Westernizing indigenous ways of knowing. In your work, have you found a better way that's more about coming up with something new that works really well for everybody?
So co-production, I mean, that's fantastic to get a question on that. So there was recently a letter to NSF written by some different organizations in Alaska, some different indigenous organizations, really asking for co-production in the research project, in the research process for the new Arctic funding stream coming from NSF. And what I've really found as far as my own work and in discussing co-production and what community-based research means is really that instead of, developing the whole grant and then going to the community and presenting and be like, can I get a letter of support for this project to be done in your community? It's really, where can you start before that? So, that whole model I gave about knowing community history, finding local contacts. So the relationship building phase of my project started about October 2016. Then I visited the community. I started my process with the community. I got a letter of support from the tribe in February 2017, which still the project wasn't developed. In July 2017, I visited the tribe and developed the methodology and research questions they were interested. I applied for IRB, then I applied for NSF. So the relationship building started in October 2016. The actual on the ground of the project was not until March 2018. So I think it's really important to think, it's a different way of thinking about things, building in that long-term commitment and relationship building that communities are really looking for. But then, you know, your work can have so much more benefit and impact. And it also helps, you know, that you won't be researching something then that the community would be like, oh, we already know that, you know? So really connecting with people to talk about the process and your interests and their interests, and, you know, seeing where you can be more in sync. Because communities aren't just interested in, you know, fixing, you know, working with their language or bringing their language back or working with their culture. They're interested in physical science stuff, you know, climate change is hitting the Arctic. We have issues with, you know, species migrations. We have issues with, you know, what is oil, the oil drilling impact to our whales if we're doing offshore drilling. So there really is fantastic ways to connect with traditional knowledge holders around these issues. And some, you know, some gateways that I look at is, you know, people approaching me or approaching the University of Alaska Anchorage or University of Alaska Fairbanks, you know, to look at, do you have connections to communities in this area? This is where I'm interested in working. And then in the Alaska context, also, we have what are called nonprofit regional corporations. And these often represent large collectives of tribes in Alaska. And they often have a public-facing person or a research-facing person. So there's a few different avenues you can kind of start working to build those local contacts and then build on that from there.
Great, thank you so much. We have another question, this one for Dr. Bowman. There is a question about what you consider to be the top 3 most pressing issues to research about the Arctic Ocean's biogeochemistry.
Well, I'm biased to methylmercury because that's mostly what I've studied in my career. But methylmercury is the form of mercury that gets into seafood. It's the most difficult form of mercury, in my opinion, to study. I mentioned that models are kind of the goal of collecting this type of data. And there are about at least 10 different models that predict the gaseous mercury and the total mercury concentration in the ocean that include the Arctic. There are 3 that approach estimating methylmercury in the Arctic. And that's because their data is just so limited. It's so hard to get. So my number 1 focus area would be understanding more about the chemical cycling of mercury in the Arctic seawater. I mentioned finding genes that are important to the mercury cycle, but we don't know how active those genes are. So looking more into the microbial species that live in Arctic seawater, how they're changing mercury chemistry, and just being better able to predict methylmercury concentrations, which is incredibly challenging in the ocean. Second research area I would say is focused on permafrost. And I know I have some colleagues in Sweden that just got funding to do this, but permafrost is estimated to store twice as much mercury as the ocean and the land and all their environments together. So there's a huge amount of mercury stored in permafrost. And as permafrost thaws, releasing mercury, we're also releasing a rich source of carbon. This is food for microorganisms. And we know that some of the genes that cycle mercury are found in permafrost. So that, I think, is a really important area to focus on. What is the fate and the speciation of mercury that's coming out of melting permafrost? And I also think it's important to focus on trophic interactions and how mercury is going to accumulate in food webs as those food web dynamics are changing. So I think a collaborative project between ecologists that are studying the food web and mercury scientists would better help us understand some of the different spatial patterns in mercury concentrations in marine mammals and seabirds, and maybe come up with an answer to that first question, how are concentrations in fish going to change as they migrate north to get to cooler waters?
Again, some additional ideas for future research and collaborations that I hope participants are taking notes on. We had one follow-up question for Dr. Gordon from Elizabeth on how to convince funders of the importance of this planning work.
All right. I didn't know if we would have time, so I was trying to address some of that question in the chat. When I actually interned at the Office of Polar Programs in 2010, and one of the things we were trying to address is this exact question: how can you get funders to fund the relationship building phase if they require a relationship to be written into the project when you actually submit the large grant application? As I ended up writing a white paper for them, letting them know that many researchers wanted this, and what can we do about this? And there is some small, eager type grant funding to build stuff, but I would say still largely it is not fundable. And even for my own work, 2016 to 2019, that's 6-9 years after, we first started exploring that. And still the majority of my relationship building phase was my own dime for sure. And so I have to say that these networks like the IARPIC, the Arctic Frost, this migration network, I feel these, by engaging with these, they're one way that kind of the funders are funding this, not funding your relationship specifically, but kind of trying to fund this broad development of relationships. So I kind of see that happening, but I do agree that is still a very difficult part. And especially in physical science work, if you're not doing transdisciplinary or multidisciplinary work and you're just applying for funding for physical science, I think that can be a very hard sell. And I think often, physical scientists giving back to the community might not even be a part or included in your grant, but just might be something that you do as a person that, helps you feel like you're doing things in a good way. So I'm sorry, I don't have a super helpful answer there.
Well, I think that's a really informative answer for what further change we can accomplish together, like things like the letter that you mentioned on co-production, maybe additional letters on the importance of planning to ensure that everyone has leadership in grants. As we are wrapping up today, perhaps one final question for both of our presenters, because we have talked quite a bit about funding, about the potential for future projects and informing future research. I know we have many early career researchers, traditional knowledge holders, practitioners on our call today. Do either of you have any advice for those who maybe are still finishing their graduate programs, are looking to apply for that first grant? Any words of support for our earlier career participants today?
I guess I'll just say, jump at any opportunity you can find, if it's volunteer, if it's temporary. For me, going to the Arctic didn't really line up. I wasn't going to be done with my PhD yet, and it was an opportunity to go to do a postdoc. But I pushed and I finished early, and I kind of jumped at that opportunity, and I had to change my plan. I had to change my path. I had to move to California months before planned. But just Yeah, search for any opportunity you can find, even if it's just volunteering. Wash bottles for someone in their lab. That's how I started as an undergrad, kind of getting into research. All of those sample bottles I showed you outside the ice tent, those have to be cleaned and it takes a lot of time. So volunteer to do things like that, that might get your foot in the door in a lab that may eventually lead to an opportunity to do bigger research projects.
And from the angle that I approach things, also talking about, instead of like more thinking of how to engage with the lab, in the more how can you engage with communities you're in. You know, most of us are living on land that was actually indigenous land at one point. So who are the communities, you know, that have that land, that still have space? Who can you engage with and just talk to about your work? And you know, one of the biggest things is, as Dr. Bowman said, being open to those changes, because, you know, you might have a research interest, but the more you start talking to a community, you see that, oh, their interest is a little more in this angle. How can I come around to, you know, approach that question? Is that something that is on the edge of my wheelhouse that maybe I can like build up some strength in? So for my work, you know, the really looking, some of the results really showed that the follow-up work that the community wanted was very physical sciencey. So a part of the research that I did when I finished was connecting that community to UAF. I was the first researcher that had ever worked in their community. So, you know, building those connections is also a great role that we can play to help continue the collaborative work.
Great. Thank you both for those wise words and a great point to finish our webinar. Dr. Bowman, Dr. Gordon, thank you so much for being so gracious with your experience, your expertise, your time today. I, again, am going to ask everyone to come together and give a huge virtual round of applause for such a fantastic start to all of our week. And you can have a fantastic end to your week as well with another Breaking the Ice Sealing webinar that is happening on Thursday with 2 more amazing presenters that will be speaking about Arctic women explorations of the past in both North America and Russia. So please join us for that, and an open invitation to anyone who would like to join and present at a future webinar in the summer and fall. We aim to be as inclusive as possible and uplift as many voices as possible. In our follow-up email, we'll be sharing a form that you can either nominate yourself or someone else to share their expertise, research, and experiences. Well, until our next Breaking the Ice Sealing series webinar that I'm sure I'll see you all at, I hope you have a fantastic start to your week, and thank you again for joining us. All right, that is all from me.